Assalamualaikum warahmatullahi wabarakatuh. Today I will present about detection and identification of pathogen and non-pathogenic bacteria recovered from duckweed Wolfia globosa during storage. My name is Ina Atira binti Muhammad Sabri, 1171506, supervised by Dr. Hanis Nadia binti Yahya.
This is my research outline. In this introduction, I want to highlight that duckweed offers a higher protein balance of essential amino acids compared to soybean. Wolfia arhiza and Wolfia globosa are the most species used in human meals, according to K. Sojanasri 1 and Cross J. Apperons. Increased fresh produce consumption and an increase of outbreak attribute to production between 1973 and 1997, according to Siva Balasingham and the other co-researchers. The presence of bacteria in plant could possibly from cross-contamination, poor handling during processing, and improper water irrigation during growing and handling the vegetables, according to WHO 2020. The problem statement in my research study is nowadays people tend to consume raw vegetables and fruits as they have known that cooked food usually lose some nutrition during heating process, according to Sentinek. Vitamin B and C can be easily lost in boiling water because it is water soluble and heat sensitive. When submerged in hot water, can leach from vegetable and fruit, according to Spritzer. The raw foods usually contain dangerous bacteria that only can destroy by cooking, according to Bell 2017. The purpose of my research is to generate information of microbial profiling of raw duckweeds by highlighting on identifying both pathogenic and non-pathogenic bacteria such as E. coli species, Bacillus species, Staphylococcus aureus, and Listeria monocyte. The objective of my research is to determine the microbial loads of Wolfia globosa during storage at 4 degrees Celsius on days 0, 3, and 6 of storage using total plate count method to isolate and identify bacteria during storage at 4 degrees Celsius on days 0, 3, and 6 of storage using 16S rRNA sequencing. To detect pathogenic bacteria during storage at 4 degrees Celsius on days 0, 3, and 6 of storage using selective media agar. The literature review is Wolfia globosa is the smallest flowering plant that does not have a root and purify wastewater, according to Fujita and the co-researchers. It contains high nutrient value and omega-3 fatty acid that could be high protein quality human food, according to Beckett. Species of the genus Wolfia are traditionally used as human food in some of the Asian countries like Laos, Thailand, and Myanmar, according to Watson. Many foods must be stored in the refrigerator and thus kept below 5 degrees Celsius to minimize the risk of bacterial contamination, according to AIFS 2016. However, there is likely to be a gradient in the temperature range that microbial life has adapted, such as Escherichia coli, which is categorized as mesophilic bacteria, can adapt and grow in cold environment, according to Caviccioli. Fruits and vegetables normally carry both pathogenic and non-pathogenic strains of bacteria, according to Jagger. The bacteria including Pseudomonas and Xanthomonas, which cause leaf spots or blight, according to Chase. The Erwinia cordovirus bacterium usually causes sore throat to many plants, according to Laram. In May 2011, hemolytic uremic syndrome outbreak is come from raw contaminated sport infect the other vegetables in salad. Meanwhile, enoki mushroom by Greenco Ltd, based in the Republic of Korea, was the source of listeriosis infection, according to CDC 2020. Firstly, sample preparation and treatments, determination of total plate count, next, determination and isolation of pathogenic and non-pathogenic bacteria, then molecular identification of pathogenic and non-pathogenic bacteria, next, data analysis, lastly, phylogenetic tree. Sample preparation and treatments. Duckweed will be grown in the several plastic basins at Pahang. The sample will be harvested and divided into sterilized seal bags of 25 grams each and will be isolated at 4 degrees Celsius for 6 days. Bacteria count and isolation of pathogenic bacteria will be conducted on day 0, 3, and 6. Determination of total plate count. Preparation of nutrient agar for total plate count. Next, preparation of peptone water for sampling preparation. Next, diluting and incubating bacteria from sample. Lastly, counting the microbial loads. Determination and isolation of pathogen and non-pathogenic bacteria. Isolation of non-pathogenic bacteria from nutrient agar, count and isolation pathogenic bacteria, preparation of bacteria stock culture for molecular identification confirmation. Next, molecular identification. Firstly, DNA extraction. Next, DNA quantification and purify. Next, DNA application PCR method. Then, DNA qualitative analysis by gel electrophoresis. Lastly, DNA sequencing. Next, for data analysis, statistical analysis will be done using Microsoft Excel 2010 and Minitab 16. Meanwhile, phylogenetic tree will be built on Cluster Omega website. Expected result for this research is both non-pathogenic and pathogen bacteria are present in Wolffia globosa. The total plate count will increase as the microbial load also increases throughout the storage process. The lab analysis will start in October 2020. The range to complete the analysis in October to December 2020. Thesis submission will be on January 2021. These are the list of the reference.